Welcome students to a continuation of the urinary or renal system. This particular lecture is going to focus on, really mostly on nephrons, the part of the kidney that actually filters our blood and makes our urine. We have about 1 million plus nephrons per kidney, so you've really your kidneys are just chock full of these suckers. They have a very kind of particular structure. They consist of the glomerular capillaries, kind of surrounded by the Bowman's capsule or the glomerular capillaries. So it's a particular capillary bed that is then surrounded by a capsule, which then, once that's kind of what filters the blood, and then the filtrate then moves into the proximal convoluted tubule, down the descending limb of the loop of Henle, up the ascending limb of the loop of Henle, into the distal convoluted tubule, and then finally into a collecting duct. And the collecting ducts actually collect urine from several nephrons, and they'll descend down through the medulla, carrying the urine into the pelvis. So the convoluted tubules are so called because they're kind of intricately kind of folded in on each other, and they kind of loop in and around each other, so they're convoluted looking, complicated looking. The proximal convoluted tubule is the one that is closer to the glomerulus, so it's in terms of like a kind of filtrate flow, so it is proximal, and then the distal convoluted tubule is kind of further away in the filtrate flow. It's more distant, so it is the distal tubule. You actually have two types of nephrons in your kidneys. Most of these are what we call cortical nephrons, wherein pretty much the entirety of the nephron sits within the renal cortex, and then you have some nephrons that are called juxtamedullary nephrons. Juxta means next to, so next to the medulla. So these nephrons, they still sit in the renal cortex, but they have these really, really long loops of Henle that descend down into the medulla. So urine formation, which is what the nephron does, it's completely, the whole process of urine formation is carried out by the nephron, is really three processes, not just one. The first process is glomerular filtration. So here you can see the cortical radiate artery, blood coming into the first capillary bed. That is where all of the filtration takes place, and then the blood leaves the capillary bed and eventually ends up in another capillary bed. The renal tubules carry out reabsorption and secretion. So reabsorption is kind of the reuptake of stuff that was in the blood that got filtered out at the glomerulus but is still useful to the body. Water, glucose, amino acids, vitamins, different ions, kind of depending on what's going on in our body at the time. All of those things will be filtered out of the blood here at the glomerulus, but they are useful, and so they are reabsorbed by the body, and so they move out of the renal tubules and into a peritubular capillary. Tubular secretion is the kind of opposite of that process. It is solutes and stuff, waste products, that will move from the blood into the renal tubules. This may be something that was perhaps too big to be filtered out of the blood up here, or something that was passively reabsorbed that we can't use, so we secrete it and try to get rid of it. So we'll start with the glomerulus and the filtration process. Filtration itself is a passive process. It's pretty much essentially just due to water pressure, kind of pushing fluids and solutes out of the capillary and into the space, the little Bowman space. It's not like a process that requires a lot of energy. Now, the secretion and reabsorption processes are kind of very metabolically intense, but filtration is really just the blood moving through the filter of the glomerulus. And the glomerulus itself happens to be a very, very high pressure capillary bed, much higher blood pressure in this particular capillary than, like, say, your typical capillary. Part of that is that it is both fed and drained by an arteriole. So the afferent arteriole leads into the glomerular capillaries. The efferent arteriole leads out of the glomerular capillaries. Because they are both arterioles, they are both high resistance vessels, which creates a pressure gradient. The other thing that creates a very high pressure system here in the glomerulus is that the afferent arteriole is wider in diameter than the efferent arteriole. So we have a wide tube for blood coming in and then a smaller tube for blood leaving out, right? Think of it almost like three cars trying to merge into one, right? That kind of creates a backup. Well, what it's creating a backup in the glomerulus is of pressure. So very, very, very high blood pressure in the glomerular capillaries compared to your typical capillary bed. That high blood pressure essentially forces fluids out of the capillary and into the surrounding capsular or Bowman's space. Now, because the glomerulus is so high pressure, there is no reabsorption that takes place at this particular capillary system, which is very unlike your kind of typical capillary in the body. The glomerulus itself has kind of a couple layers in terms of kind of surrounding it, in terms of the kind of the wall structure. In particular, it has what we call these podocytes or foot processes, and they make what are filtration slits, essentially kind of the holes of the sieve, if you think of it like a colander that you drain your pasta in. So in order for a solute to be filtered here at the glomerulus, it has to be small enough to fit past these filtration slits. And not everything is. And that is why we need that kind of secretion process. So there are three types of pressures that affect glomerular filtration. One of those is what we call the hydrostatic pressure in the capillary itself. It's basically the blood pressure of the capillary. And again, because of the arterioles, it's very, very high compared to a typical capillary. That is a outward force, kind of pushing fluids and solutes out of the capillary and into the surrounding Bowman space. As the Bowman's capsule fills up with filtrate, well, that filtrate exerts its own hydrostatic pressure, water pressure, pushing fluids and solutes back into the glomerular capillary. So that's an inward force. We also then have an osmotic pressure in the glomerulus itself. Because proteins are not typically small enough to fit past the filtration slits of the podocytes, all of our plasma proteins essentially stay in our blood, and they act like a vacuum, kind of pulling and sucking water and fluids back into the capillary. So we have one kind of outward force, two inward forces. If you subtract the inward forces from the outward force, you still end up with a positive filtration pressure, meaning filtration is going to happen. At no point does reabsorption occur here in the glomerulus. It's all filtration all the time. Now we can change the rate of filtration. We can make filtration happen faster or slower, but it's always going to be filtration here at the glomerulus. in terms of the renal tubules, this is where we're going to find the reabsorption and secretion taking place. And most of the reabsorption that takes place in the renal tubules takes place in the proximal convoluted tubules. So about 65% of your filtrate volume is reabsorbed here in the proximal convoluted tubule. Again, anything useful that was filtered out, but we want to keep. Water, bicarbonate, any kind of nutrient, some sort of maybe sodium or something like that to, help us heart, brain, nerve function. Pretty much anything that your body can use that we don't want to lose that was filtered, your body will reabsorb. And so the proximal convoluted tubule actually has a ton of little finger-like microvilli to create more surface area to allow more reabsorption to take place. Now, the loops of Henle tend to reabsorb salts and water and play a role, particularly the loops of Henle for the juxtamedullary nephrons in kind of regulating the concentration of our urine. And then the distal convoluted tubule can do, aldosterone kind of stimulated secretion of potassium, reabsorption of calcium and sodium, again, hormonally based. Most of the stuff that the distal convoluted tubule either reabsorbs or secretes is usually some sort of hormonal stimulus for kind of turning that on. There are two other types of capillaries associated with our nephrons, depending on whether we're talking about a cortical nephron or a juxtamedullary nephron. The cortical nephrons, were the ones that sit almost completely in the cortex, they have peritubular capillaries around their proximal and distal convoluted tubules. And the peritubular capillaries are very, very low pressure capillary beds that are reabsorbing nutrients and fluids. The vasa recta capillaries, also low pressure, but they are not around the convoluted tubules, but they are around the loops of Henle of the juxtamedullary nephrons, and incredibly, incredibly important for kind of regulating the concentration of our urine, right? So if we're really dehydrated because it's 85 degrees out and we've been, working outside in the hot Georgia sun, then when we go and we void our bladder, when we urinate, we tend to have a very small amount of very dark urine. It's very concentrated. Our body is trying to hold on to as much water as possible so we don't get dehydrated. Conversely, if you're inside in a temperature controlled room and you're drinking copious amounts of water, if you go and urinate, you're going to have a larger volume of urine. It tends to be kind of pale yellow, so it's not as concentrated. And one of the things that helps us do that are these vasa recta capillary beds. The other little bit of kind of nephron anatomy we have to discuss is something called the juxtaglomerular complex. Again, juxta means next to, so we're talking about this complex of cells that are next to the glomerulus. And it's really two kind of components, what we call the macula densa and the granular cells. So the granular cells are actually found in the wall of the afferent arteriole, and they are pressure receptors. If your blood pressure in the afferent arteriole is too high, the granular cells sense that and cause the afferent arteriole to vasoconstrict so that we don't have too high a pressure kind of getting into the glomerulus, can restrict the flow a little bit so that we essentially don't blow out our capillary and start bleeding internally. The macula densa are salt receptors, they're chemoreceptors. They are found in the wall of a portion of the loop, the renal tubules that kind of loops back next to the glomerulus. And if the salt concentration in the filtrate is too high, it indicates that the filtrate is moving too quickly through the tubule system and there isn't just enough time to reabsorb kind of needed, necessary things. And so the macula densa kind of communicates that. That then causes the afferent arteriole to vasoconstrict and get smaller, which slows the filtration rate. So filtration is happening slower, which means the filtrate flow is slower, and that allows the renal tubules kind of enough time to reabsorb the salt so that we kind of have a proper concentration of it. So if we look at the filtrate flow, it is created by the glomerulus, right? That's where the filtration happens. The filtrate then fills up the glomerular capsule and then into the proximal convoluted tubule, down the descending limb of loop of Henle, up the ascending Henle loop limb, and then into the distal convoluted tubule, into the collecting duct. Those collecting ducts are going to descend down through the renal pyramids, and then at the apex of the pyramid, there's going to be a little tiny hole that will allow the urine to move into the minor calyces, which merge into a major calyx. The major calyces then merge with the renal pelvis. The renal pelvis is continuous with the ureter, which takes the urine to the bladder, and then it is the urethra that gets the urine out of the body. That process of voiding your bladder, urination, is called micturition. And the kind of stimulus for urination is when the bladder wall gets kind of stretched out because it's full of urine. That initiates a reflex. And three things have to occur. The autonomic nervous system is going to cause the contraction of all of that muscle in the wall of the bladder. The autonomic nervous system will cause the internal sphincter to contract and open, and then you have to voluntarily choose to relax the external urethral sphincter in order to void your bladder at an appropriate time. Now, infants, they do not have the capability to do that, right? That's why they have to wear diapers all the time. They can't void their bladder at an appropriate time. They just kind of do it whenever it happens. It's reflexive. They don't have the ability consciously to control the external sphincter, but they actually don't also have the kind of brain development for storing urine. So they're in the brainstem. You have centers that say, hey, it's time, that stimulate urination and that stimulate urine storage. And those centers won't develop typically until an infant's about 2 or 3 years old. And so that's why they just can't control it. And it's not typically till they're about 2 or 3, and then you can potty train your kid. The other thing that you guys are going to do in lab is actually exercise 41, but it's basically a urinalysis. And urinalysis is super useful because if you have something in your urine that is not supposed to be in your urine, it could indicate and can give you some indication, some kind of, well, if it's this, it might be this that's causing that. What is wrong with your patient? The other thing that's really neat about urinalysis is it's super easy to do. You're basically going to get a little cup of simulated urine, stick the little test strip in it with all of the chemicals on it, with all of the kind of indicators. The indicators will change color, and you'll be able to read the stick and determine a whole host of things in terms of what is in the urine. Normal components of urine are things like water, our nitrogenous containing compounds, urea and uric acid, creatinine, which happens to be a waste product of our body's metabolism, like urea and uric acid, and then any number of ions. You might find bicarbonate ions, you might find magnesium ions, you might find sulfate ions. That's all perfectly normal and can vary essentially with the body's needs. Now, abnormal things can indicate that there might be a problem with your patient. So for instance, if your patient has glucose in their urine or ketones in their urine, it could indicate that they have hyperglycemia, they have diabetes, and don't have a control on their blood sugar. And they have too much sugar in their blood, and when it gets filtered out, there's not enough it can't get all reabsorbed, and some of it shows up in the urine. If you have proteins in your urine, that typically indicates that there is damage to the filtration membrane at the glomerulus. Proteins are typically too large to be filtered out, and so if they're showing up in their urine, it could mean that the filtration membrane has been damaged. If you have blood in your urine, it could indicate that you have some sort of infection or a kidney stone or cancer. There's a whole host of reasons why blood can show up in your urine. If you have nitrates in your urine, that might indicate a bacterial infection. Many bacteria, as a part of their metabolism, make these nitrates, and so if they're showing up in your urine, it could be a way of saying, hey, you've got a bacterial infection somewhere along your urinary tract. If you have bile in your urine, could indicate liver damage. Bile is made by the liver as part of our digestive processes, and if it's showing up in your urine where it's not supposed to be, it could perhaps mean that your liver has been damaged in some way. And then if you have white blood cells, leukocytes, showing up in your urine, could be that you've got a UTI, kidney infection, some sort of infection that's causing leukopoiesis, higher numbers of white blood cells, and they're essentially showing up in your urine when they really aren't normally in there. So you guys will do a little urinalysis test in lab this week. Like I said, pretty easy. Stick the strip in the urine, read the results. Takes about 3 minutes. That is the end of the renal system.